The first objective, which is to describe the various properties of graded potentials, including the direction of change in a potential, the magnitude of change. We're going to also talk about temporal and spatial summation. And then we're going to speak about how graded potentials can then trigger an action potential. So first off, we've got two general types of electrical signals. We've got graded potentials and we've got action potentials. And you can kind of think of graded potentials as weaker forms of action potentials. They're not as significant, they're smaller, right? They're not as forceful or as meaningful, and they're not as sustained. So they don't communicate over long distances. In fact, they're just kind of just blips in the membrane potential, and they only last for very short duration, for very short distances, and then they just kind of dissipate. Action potentials, on the other hand, are larger, more influential, more meaningful membrane potential changes that are sustained. They're going to be maintained for a long duration of time, as not a long duration, but for a fixed duration of time, as well as they're going to be maintained over a long distance physically as well. So the general difference here between graded potentials and action potentials is that we've got a small change in the membrane potential as far as graded potentials that do not get to this threshold value, and I'll talk about this threshold value in a bit, whereas an action potential is larger, it is sustained, and it's more meaningful of a change that goes beyond this threshold value and is actually able to travel for a long distance of time. Okay, so let us first look here at different types of potentials. And while doing so, let's also review the general structure and sort of the components of a neuron. So we're looking here at various types of potentials. And I encourage you to reference back to that table that I pointed out at the beginning of our previous packet that basically compared the different types of potentials, so equilibrium potentials, resting membrane potential, graded potential, action potential, so that you're clear as far as the description and being able to differentiate between these different types of potentials as well. So let's draw our neuron. So if you remember from one of your prerequisites, what a neuron looks like. So we've got this neuron here. This is the soma. We've got the length of the neuron, which is the axon here. And then we've got the axon terminal. So let's kind of label these out here. So here we've got the soma, or the cell body. Here's our soma. We've got, let's make these a little bit more defined. We've got dendrites that are these sensory fibers projecting from the soma. These are picking up signals from other neurons. So these are our dendrites. We've also got the axon hillock. So the axon hillock is pretty much the neck of the neuron. And this is going to be a really important region as far as determining whether or not an action potential is fired. So this is the axon hillock. We've also got the axon terminal. And then we've got the entire length here, which is the axon itself. Now I want to point out, we've also got other neurons communicating to this neuron here via the soma, or the cell body. And so we can describe the area, and we'll talk a little bit about this in the next packet, but we can describe the area of communication between two neurons as the synapse. So this communication here where this neuron speaks to this neuron, this is called the synapse. And so if we're looking at the synaptic communication, we can also refer to this neuron as the presynaptic neuron. And this second neuron is going to be the postsynaptic neuron. Okay. So this is how neurons speak to each other. They do so via the synapse. And this is what that action potential is all about. It's a form of energy that one cell can use to speak to or communicate to another cell. Okay. Let's look at some of the structures that are seen or the channels that are seen in different regions of the neuron. So at the dendrites, we're going to have ligand-gated channels. Ligand-gated channels. And if you think about what the word ligand means, it means something that binds. And so a ligand-gated channel will only open when some specific structure or molecule binds to it. So ligand-gated channels. At the axon hillock, we've got voltage-gated channels, so similar, but here, a specific voltage needs to be met in order for this channel to open. So these are voltage-gated channels. Oops. So voltage-gated channels here. Along the axon, we've also got voltage-gated channels as well as leak channels. As far as the soma or the cell body, we've got ligand-gated channels here as well. And then lastly, at the axon terminal, we have an entirely different channel. These are calcium channels. Calcium channels. Okay. So we've got these various types of channels throughout different parts of the neuron. And so now we can really set the stage for talking about the different types of potentials. So graded potentials are going to be found anywhere along the neuron that's proximal to the axon hillock. So here is where we see these graded potentials. So here we have various small, less impactful changes to the membrane potential that may or may not get to threshold. And so they're going to be small, not as sustained, and will determine or decide at the axon hillock whether or not those graded potentials, when we add them together, if they are meaningful enough to signal an action potential, then we will do so at the axon hillock. And so an action potential is going to be propagated beyond the axon hillock. So beyond the axon hillock, we no longer see graded potentials, but if we meet the requirements, we then see the development and the propagation of an action potential. Okay. So really important point to make, graded potentials are before the axon hillock, so really throughout the dendrites and the soma and cell body, whereas beyond the axon hillock, if we meet that threshold requirement, what we see are action potentials that are propagated down the length of this neuron, and then that changes to chemical energy at the axon terminal. Okay. All right. Any questions? Any questions on this before I move on? Okay. I'm going to make one small note here, which ties back to something we mentioned before, that we've got 25 times more potassium leak channels than sodium leak channels. Okay, we want to bear that in mind as we speak about potassium and sodium movement in terms of an action potential. Okay, so let's move along here. Let me clear this. So just to kind of reiterate here, the difference between graded potentials, again, they're initiated by some type of stimulus. So this stimulus can be very different. It can be a physical or external stimulus, such as you touching a hot stove, which is a tactile stimulus, or maybe auditory sensory information, visual sensory information, olfactory sensory information that is being inputted. It can also be a chemical stimulus, which is some type of neurotransmitter that is communicating to this cell. So these are the types of stimuli we mean when we talk about a graded potential being initiated by a stimulus. The change is small, so not very meaningful, right? Doesn't actually get beyond that threshold that I'll talk about. It can vary, so it can be different gradations. It's gradable then, it can be different values as far as the actual membrane potential change. So we can see a small graded potential here. We can see a slightly larger graded potential here. It is spread by electrotonic conduction, which is a fancy way of saying electrical energy, right? That negative and positive across the cell membrane, which creates electrical energy. And then it is decremental, which means that as it travels, it loses its magnitude, or it pretty much dissipates as it travels further away from the site of the stimulus. So we can see here, this is where that stimulus was first appreciated. And then as we move further away from that site of the stimulus, it kind of decrementally decreases in terms of the amplitude, or dissipates. Graded potentials are really about determining whether or not we have enough of a membrane potential change to get us to threshold so that we can fire an action potential. So it's really about adding or analyzing or determining how strong these smaller membrane potential changes are and whether or not they get us to that threshold value. And so beyond that point, we must fire an action potential as long as we pass that threshold value. And so we can have different types of changes, different types of graded potentials. They can be depolarization events, which are positive changes to the membrane potential. So the membrane potential becomes more positive, which is reflected by an upward stroke in terms of this graph here. Or they can be inhibitory events or inhibitory changes or hyperpolarizing events that actually make it harder to fire an action potential and move us further away from that threshold. So here is a hyperpolarizing event or an inhibitory change of the membrane potential. And here is a depolarizing event or an excitatory change of the membrane potential. And so all of these small changes, they can be positive or negative. And at the axon hillock, we pretty much kind of add all of these small inconsequential changes together and determine whether or not we've got enough strength that gets us to that threshold value so that we can fire an action potential, which will be propagated down the rest of the axon.